that my word?
Okay, so we first, we left off talking about DNA replication. Y'all listening? Y'all listening? Yes? Okay. We started off talking about DNA replication and eukaryotic cells. And so we're going to replay this video. And, well, first I'll give you a little brief intro about what's happening in DNA replication. Then watch the video, and then we'll move on to the last part of the lecture. So remember, in DNA replication, all we're doing is we're making a copy of our double-stranded DNA. And so in order to do that, there's going to be several different enzymes that are going to be involved. First thing is the helicase. It's going to come in. It's going to unwind that double helix. And then you've got those single-stranded binding proteins stabilizing the unwound helix. And then you have primase come in, which is going to generate that short little RNA primer. And DNA polymerase is going to come in, bind to the primer, and start making new DNA. And then after that, you're going to have DNA polymerase 1 come in, and it's going to cut out the RNA primers and then fill in the gaps where the RNA was and replace it with DNA. And then finally, you're going to have the ligase come in, and it's going to glue all those different pieces of DNA together. Remember, we have the lagging strand and the leading strand. The leading strand is synthesized one continuous piece of DNA, and the lagging strand is made in these Okazaki fragments. So you have to have DNA ligase come in and kind of glue all of these Okazaki fragments together. So we'll watch this video real quick, and then we'll move on.
DNA replication begins at a specific point in the DNA molecule called the origin of replication site. Initially, the enzyme helicase unwinds and separates a portion of the DNA molecule, after which single-strand binding proteins react with and stabilize the separated single-stranded sections of the DNA molecule. The enzyme complex DNA polymerase engages the separated portion of the molecule and initiates the process of replication. DNA polymerase can only add new DNA nucleotides to a pre-existing chain of nucleotides. Therefore, replication begins as an enzyme called primase assembles an RNA primer at the origin of replication site. The RNA primer consists of a short sequence of RNA nucleotides, complementary to a small initial section of the DNA strand being prepared for replication. DNA polymerase is then able to add DNA nucleotides to the RNA primer and thus begin the process of constructing a new complementary strand of DNA. Later, the RNA primer is enzymatically removed and replaced with an appropriate sequence of DNA nucleotides.
DNA polymerase 1.
Because the two complementary strands of the DNA molecule are oriented in opposite directions, and the DNA polymerase can only accommodate replication in one direction, two different mechanisms for copying the strands of DNA are employed. One strand is replicated continuously toward the unwinding, separating portion of the original DNA molecule, while the other strand is replicated discontinuously in the opposite direction with the formation of a series of short DNA segments called Okazaki fragments. Each Okazaki fragment requires a separate RNA primer. As the Okazaki fragments are synthesized, the RNA primers are enzymatically replaced with the appropriate DNA nucleotides, and the individual Okazaki fragments are then bonded together into a continuous complementary strand.
Okay, questions about replication before we move on? Anybody? Okay. And by the way, I posted the link for this video, the YouTube link, on an announcement on Blackboard. So if you want to see this again, you just click on the link and it'll take you right there. Next. Yes, Daniel.
Isn't there another name for the Okazaki fragment?
Absolutely not.
Wait, what's ligase?
Ligase is the enzyme that glues the Okazaki fragments together. So if you ligate something, you're usually tying something together. So ligase is going to join those Okazaki fragments together.
Okay.
Other questions? Okay. So the rest of the lecture is going to focus on what happens after DNA replication. And so before we move on, I want to tell y'all about how we package all of our DNA into our cells. I think I told you about the stat of how much DNA we actually have, 1.8 billion kilometers worth of DNA. So a crap ton of DNA, right? Well, we got to get it all into our cell some way. And so the way that we do this is through a series of chromatin organization. And so there's various different levels, but if you start just with the double helix, so you unwind all the DNA and you're just left with the double helix. The way that that helix gets condensed, or in other words, tries to package itself into a smaller size, is it wraps around, the DNA will actually wrap around one of these things called a nucleosome. And a nucleosome is going to be a series of 8 proteins called histone proteins. And so when you have those 8 histone proteins come together, they make kind of like a little cube. That's the nucleosome. And so what's going to happen, your DNA is going to wrap around it like 2.5 times. And then it'll be a little gap, and then it'll wrap around another nucleosome, then another gap, and it'll continue wrapping around these little balls of histones. And so then, if you can imagine that we've taken all of our double-stranded DNA and we've wrapped it around these nucleosomes. Now what we're going to do is we're going to start making coils of all of those nucleosomes. So what's here is now that's nucleosome, 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 so they're all packed in there super, super tight. And so those nucleosomes will start to be coiled, and then these coils will start to coil on top of the pre-existing coils, and then those coils will continue to coil up, and you just end up upon coils upon coils upon coils of DNA, and so you get this super condensed version of chromosomes. So remember when we said we have to replicate our DNA? Well, when we replicate it, it has to condense before we can replicate it. And so this is one of those ways that we can condense and decondense our DNA. And so anyways, that's how we get from the super, super long strand of DNA into all fitting within one nucleus. And the analogy I like to use is, some of y'all are old enough to remember before we had wireless phones. The phones that have like the little curly cord, and if you sit there talking on the phone, you play with the cord and like twist it, and you twist it, and you keep twisting it, and it kept getting like more and more and more condensed. Same sort of idea. So it's coils upon coils upon coils, and that's how you get all this DNA into this one tiny little package. Okay, so we've already talked about what happens during DNA synthesis. And so there is something that's called the cell cycle. And the cell cycle is there to regulate when a cell is dividing. Because your cells are not dividing all the time. They're just dividing every once in a while. Like most human cells divide maybe like once a day. So it's not like it's dividing, 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 dividing. And so you have this kind of internal clock, which is the cell cycle, which tells the cell, okay, you can go ahead and divide. And then once the cell divides, then it'll leave the cell cycle and go back to being its just normal cell. And so this cell cycle is made up into 4 different phases. We've got G1, S, G2, and M phase. We've already talked about what happens in S phase. S stands for synthesis. So what did we just talk about synthesizing? DNA. So this is where we're going to replicate our DNA, is in the S phase of the cell cycle. Which makes sense because when you start with one cell and you end up with 2 cells, the process of cell division, if this has 1 chromosome, we got to distribute it evenly into both new daughter cells. So you have to make a copy of it so that once it divides, each daughter cell now has a full set of genetic information. So before we divide our cells in half, we have to make a copy of our DNA so that the new cells that we're making have all of the DNA of the original cell. Does that kind of make sense? All right? So if I was going to distribute a whole bunch of tests to y'all on your exam day, I have my test. In order to get every single one of you a test, I have to make copies of it, right, before I distribute it. Same sort of idea. I have to make a copy of my DNA before I sort it into 2 different cells. so that after cell division, you end up with the same genetic information in each cell. Right? Makes sense? Logical? Yes? Okay. And so when a cell is not dividing, and by the way, most of the time, your cell is only in the cell cycle about 10% of the time. So that means 90% of the time, it's not even in the cell cycle. So when it's not in the cell cycle, that 90% of the time, it's in a stage called G0. That G0, all it is just a symbol saying that the cell's not dividing. So if the cell's in this G0 stage, it's not dividing, it's not in the cell cycle, it's not replicating DNA, it's not doing anything other than just being a cell, doing its normal daily cell stuff. And so one of the other more important phases of the cell cycles is M phase. Now M phase is going to be made up of two different processes. The first one is mitosis. Mitosis is the process of this actual cell division. And then mitosis is going to be quickly followed by a process called cytokinesis. So the definition of mitosis, this is division of the DNA, right? We're making a copy of the DNA and then we're sorting it into two different cells. That's what mitosis is doing. And so after you sort the DNA, then you've got to sort the rest of the cell, the cytoplasm and all that stuff. Well, that is cytokinesis. So cytokinesis, cyto for cytoplasm, kinesis means movement. So cytokinesis is basically the division of the cytoplasm. And so in M phase, it doesn't stand for mitosis because it's going to have mitosis in it, but it's also going to be followed quickly by cytokinesis. So mitosis and cytokinesis are both in this M phase that occurs right here. And then the other two phases, these G1 and this G2, those are just kind of growth phases. Nothing really special happens there. You know, the cell's basically just kind of getting ready, it's growing, it's doing its normal cell thing. It's just kind of like a nap or something. It's on hiatus. And so when a cell enters into the cell cycle, so it receives some sort of signal, it's like, okay, I need to make a copy of myself. So this cell will leave the G0 state, and it'll jump in to the cell cycle. And it's not like the cell cycle is a physical thing. You know, it's just kind of an internal clock that's regulating the process of cell division. And so when it knows that it needs to go into cell division, it'll, enter the cell cycle and enter into this G1 phase, this growth phase. And again, that G1 phase, it's just there, the cell's doing its normal thing. It's kind of getting ready to start preparing to replicate your DNA, because that's the very next phase in the cell cycle. So when a cell knows it needs to divide, it'll leave G0, enter into G1, normal cell thing happening, and then from that point, it'll go on to S phase. And S phase is a synthesis. This is where you're making a copy of your DNA, because you have to make a copy of it before you can distribute it to the new cells. And so that's what's going to happen in DNA synthesis. So that's what we just finished talking about in the last, or the first part of the lecture. And then after you make a copy of your DNA, then you're going to go through another growth phase. Same thing, nothing really special going on here. But then once it leaves into G2, then it'll go into M phase. Then it will enter, and within M phase, again, this is divided into mitosis and cytokinesis. So mitosis will happen first, and mitosis has its own group or subset of steps that we'll talk about eventually. And then once the DNA actually gets divided, then the rest of the cell actually gets divided through cytokinesis. And so at that point, once you finish M phase, the cell's already made a copy of itself. So now the cell will leave and go back to G0. So when a cell goes through the cell cycle or undergoes cell division, it'll enter in G1, it'll go around once, and then once it's made a copy of the cell, it'll leave and go back into the G0, or the undividing stage of the cell. We okay so far?
So it starts at G1.
Starts at G1 and it just kind of goes around. So it enters in G1, it grows, it's normal, makes a copy of its DNA, it grows some more, and then we divide our DNA, and that's closely followed by the division of everything else of the cell. And then it'll leave the cell cycle and go back into being just a normal cell, just not replicating. We okay with this so far? Okay. All right, so we've kind of already discussed the different roles, but again, these 4 different stages, you've got the G1, the G2, and the S phase. The G1 and G2 are kind of these just growth, where the cell's getting prepared to go on to the next stage, whether it's S phase or M phase. And then the G0, this is this non-replicating stage. And so the whole purpose of the cell cycle is to regulate when we go into mitosis, when we go into the actual division of the cell. And so when our DNA is actually being divided, there's actually an apparatus or a complex of multiple different things that are working in unison in order to separate these DNA into other sides, or into different daughter cells, into the new cells that you're making. And so this apparatus that's going to be used is called a mitotic spindle. And so remember we talked about different types of cytoskeleton and microtubules? Well, one of those are those big, thick like cable type things that help support the cell. Well, these microtubules, that's what all these purple things are. And so these microtubules actually help and assist with separating the DNA from one another. So if we have a cell, here's my cell. It's going to enter in the cell cycle. In G1, not really much is going to happen. And let's just say this cell has 2 chromosomes, chromosome 1 and chromosome 2. So after G1, then it's going to go into S phase, right? And so in S phase, this is where we make a copy of our DNA. And then it'll go through G2, and then once it gets into M phase, you're going to actually be separating these copies to opposite sides of the cell. And the way that we're separating those copies from one another is through this mitotic spindle, which is made up of all of these microtubules. And so these microtubules, they're going to be emerging out of this structure called the centrosome. The centrosome basically is in charge of making microtubules. And so the centrosome is making all these microtubules, they're going out and they're attaching to one side of each copy. So after S phase, you end up with 2 genetic pieces of DNA. So before S phase, just imagine it being 1 copy of DNA. And then after you go through the S phase, then you end up with your second copy. So once you copy the DNA in S phase and you make it back into mitosis, mitosis is now going to divide those 2 copies from one another. Because when you make a copy of your DNA in S phase, you're literally just making a clone, a genetic clone of DNA. And they're attached together at the centromere, which is this region holding these 2 together. So now that we've replicated it, now we're going to go ahead and separate them into opposite cells. And so that's helped out through this through these microtubules. And these microtubules, they'll actually grow out of the centrosome and they'll attach. So these microtubules will attach to this, copy of the DNA. And these microtubules will attach to this copy of the DNA. And then what's going to happen is these microtubules are going to contract. They're going to shrink. They're going to get smaller. And when they start getting smaller, going back into their respective centrosome, that's the force that's actually going to separate those copies of DNA from one another. And so when we're talking about, actually separating these molecules of DNA, we're talking about the spindle apparatus, which is this thing right here. And I know this picture is kind of hard to see, but this is an actual picture. What they did is they took fluorescent dyes. They stained the DNA in blue, so you can see all these chromosomes right here. And then they stained all the microtubules in purple. And so this is showing the centrosome here and the centrosome here, and then all these microtubules coming out and attaching to each copy of DNA. And then when those microtubules start to shrink, it physically pulls those copies into different cells. So at the end of the cell cycle, in M phase, you end up with 2 cells that were genetically identical to the original cell. So All we're doing in DNA, or in cell division, you're making a copy of your DNA, and then you're distributing one copy to each new cell that you're going to be creating. And so when you get to mitosis, there's going to be 5 different stages that occur. And this is kind of a written version of what's going to happen. To save a little time, I'm going to just go through the pictures so you can actually see what we're talking about during all of these phases of the cell cycle, or all these phases of mitosis. Okay, so we said that in mitosis, this is where we've already made a copy of our DNA because we've already gone through S phase, right? So once we start mitosis, we're already starting with the copied DNA. The DNA has already been copied. And so one of the first things that's going to happen is, because before this cell goes into mitosis, it's still all kind of loosey-goosey. It's not very condensed. It's just kind of all over the place. So in mitosis, because we're moving around these pieces of DNA, it makes sense to move them in smaller packages than trying to move this whole big amorphous blob of DNA. So the first thing that's going to happen once you enter into mitosis is the DNA is going to condense. And so it's going to go from this kind of, loosey-goosey type of DNA that's just real stringy. It's going to condense until you can see these tight little chromosomes. But they're not going to be arranged in any particular. You can just see the dark staining chromosomes compared to here where you can't really see, you can't distinguish the chromosomes from one another. So that first step is going to happen in prophase. In prophase, that DNA is going to condense. It's going to get smaller, it's going to shrink up and get really, really tight. So that's what's happening in prophase. Also what's happening in prophase is you start to get your spindle apparatus